genomic research on viral tolerance and disease resistance.
Hi, Mike. Just for those following along on YouTube, if anyone's watching, we're about 20 minutes behind schedule, so we're about one presentation behind. So if you're looking towards the schedule, kind of offset by about 20 minutes, although we may catch up. So, thank you.
Okay, thank you very much. Did you just wind this up? Sorry. Yeah, I'm going to change gears slightly. So I'm going to be talking about a collaborative project that involves some work outside of Canada and also using some genetic data. Yeah, so I'm just going to start off by giving a bit of background information with the association between bats and some pathogens, including coronaviruses. And then I'll finish off with a couple of examples of some projects that we've done using the frozen tissue collections at the ROM. Yeah, so people have done studies looking at pathogens and they found that not only bats, but primates and rodents also seem to have a lot of pathogens reported. But this particular study in 2017, they also tried to correct for where papers were published, whether it was based on wild animals or domesticated or captive animals. But they still found that bats do seem to have a higher proportion of zoonotic viruses. So there is a connection of bats with a lot of these diseases, but I think the interesting part is that most of the bats seem to be healthy. So I guess that's really one of the questions that people want to find out is why are bats okay, but when people get a lot of these diseases, we get sick, and sometimes it can be fatal as well. Okay, so the connection, this didn't happen too long ago, and I'm sure most people remember the connection in the last global pandemic. And again, I mentioned that there was, bats are considered a natural reservoir for a lot of these coronaviruses. And one genus in particular, the horseshoe bat, genus Rhinodolphus, it has the closest or the most similar coronavirus to the one that, the SARS-CoV-2 that caused the COVID-19 pandemic. But it's different enough. So it has about 4% sequence difference in the RNA. SARS-CoV-2 that caused COVID-19 didn't come directly from a bat, went through an intermediate host, which there's still a bit of debate on what that intermediate host was. But for sure it didn't come directly from bats and humans. Okay, so for my particular presentation, I'll be talking about two projects that I worked on. The first one is actually on the right. I'll talk about a project that sort of tackled it from the virus perspective. What viruses are found in some of the bat frozen tissue collections we have in the ROM. And then the second part, I'll end off with using those tissue samples to actually get the genomes of these bats, the horseshoe bats, to find out, basically we sort of zero in on the immune system genes to find out why bats seem to be asymptomatic, whereas humans, we get sick when we get a lot of these coronaviruses. Okay, so a bit of background information first on the ROM. So over the last more than 3 decades, we've been doing field work pretty much around the world. Obviously concentrated in some areas more than others. So the green highlighted countries are some of the places that we've been to over the last 3 decades. And this just gives an idea of the tissue samples that we collected over the years, starting back in 1988, I believe. And just a very quick summary of concentrating just on bats. So as far as, for bats in general, throughout the world, there are 21 families of bats. So we have tissue samples that cover 15 of those 21 species, 21 families of bats. But as we get lower down the taxonomic level or scale, so we have just over half of the genera, the 220 genera of bats. But when we look at our tissue samples at the species level, we have about a quarter of the 1,400 plus species of bats. So we have a little bit better coverage at the higher taxonomic levels, but as we go lower down the species, we don't have as much. So I usually use this slide as a way to ask people if they work on different species around the world to help sort of contribute to some of these genetic studies that are trying to sequence the genomes of different species of bats. But one of the key things about our collection is that since the 1980s, we've been freezing tissue samples in liquid nitrogen. So this is very, very cold, so minus 196 degrees Celsius. So basically that's the instantaneous freezing. So that is the key to preserve, to best preserve DNA, not just DNA, but also the RNA. So the RNA is a little bit more unstable. So freezing in liquid nitrogen is key for looking at RNAs and viruses and stuff like that. But of course, there are some trade-offs. It's not always easy to find liquid nitrogen in a lot of the countries, especially when we have to go to remote areas. So you have to bring in that white liquid nitrogen tank. So sometimes it's not only difficult to get, but it's also hard to bring it into some of the areas that we want to work in. And so a lot of times we want to work in remote areas where there's a lot more species diversity as opposed to just working in urban areas. So you get more species diversity in general if you go deeper into rainforest areas like that. So just again, to say that freezing things in liquid nitrogen is the best way to recover DNA and RNA. So we've been doing this, freezing samples in liquid nitrogen for over 30 years. So actually most of the research that we do at the ROM is looking sort of phylogenetic studies, looking at evolutionary relationships, within not just bats, but in my case, mammals in general. So this is just one example of looking at relationships within a genus of bat, either the mastiff or free-tailed bats in the genus Molossus. But then sometimes there are things that we find that don't correspond to anything that we know. So sometimes we have to describe things as new species. So this is what this new species, Molossus fentonii. So this was named after Brock Fenton. I'm assuming that most of you have probably heard or know of him. So that's one of the original purposes of saving tissue samples and collecting specimens at the ROM is to look at species diversity, but also evolutionary relationships. But more recently, we found out that they're also good for viral studies and also genomic studies, which is the main thrust of this particular presentation. So this looks at, so the first part is a collaboration with some researchers at Western University. Basically, the first idea was to see if we could find and characterize coronaviruses, in particular the SARS-CoV-2. So the idea originally was to try to develop vaccines for different types of coronaviruses so we can sort of be ahead in the curve. If there is another outbreak, that we prevent it from becoming another pandemic. So this particular project utilized tissues that go more than 30 years back. And it represented about 900 specimens from about 100 species of bats. And it actually made a bit of news, and this was just a coincidence, Dan, that shout out to your network, but that was just pure coincidence. It was actually picked up by a few other sort of media outlets. So this happened basically at the height of the pandemic, so it was sort of big news back then. Still is. So I'll just quickly, so I mentioned that the main purpose was to look at coronaviruses, but we didn't find any coronaviruses in any of those tissue samples. And then when we sort of look back and we say, well, why is that? Well, COVID-19 was a respiratory disease, so we didn't actually have one. We had other tissue samples of liver and kidney, but we didn't have lung, so coronaviruses weren't picked up. So this is another example of sometimes, the main objective of the research doesn't work out, so you got to sort of pivot. So we started screening for other types of viruses, and then found out that the paramyxoviruses seem to be prevalent within some of the tissue samples. Not prevalent in a lot, only about 3.6% of the samples. But when we looked at it more closely, there were some species that had higher rates, such as this bent-winged bat. And the horseshoe bats also had the most numbers, so not the highest percentage, but the most numbers of samples with this particular virus. And then the map on the right just shows some of the geographic sampling that we had. Now just quickly go through, so what we found out that most of these paramyxoviruses were actually new or novel sequences, so these were unknown viruses, so not only are we describing unknown species of bats, the host, but also unknown species of viruses as well. So 20 of the 23 viruses were previously unknown. And most of them were in sort of an orange group. And then we picked up some novel ones as well in different groups of bats. So that, these are in the old world nose bats, and so that light blue color. And then the dark blue is another old world nose bat that had another unique undescribed species. But most of them came from that orange cluster. But you can see that it actually may not be able to see the wording there, but they represent several different genera. So that orange group of viruses, they were found throughout a lot of different species, but also genera of bats as well. And also there were some found in the horseshoe bats.
Bat folks just like you in Ontario.